Good morning, everybody. Happy Friday. All right, actually, I don't want to share screen. I want to get going on codominance. So, achy legs. Happy it's Friday. Okay, so this is where we left off on Wednesday because we ran out of time. So essentially, what I want to kind of use blood types to explain is how you can express two different phenotypes at the same time, right? And it's also really fun to talk about because it's something that you can relate to yourself as well, and you can also start working out, genotypes of your parents and stuff, right, and siblings based on their blood type. Okay, so essentially we've got 4 different blood types, ignoring rhesus factor: A, B, AB, and O. But we only have 4 different alleles. I'm actually going to spread these out a little bit. So the genotypes A and B are both dominant alleles. So you can have homozygous dominant A or homozygous recessive. And that, oops, let me just leave a space in there, will get you type A blood. We'll get into the mechanism in a little bit behind all this. It's really interesting. B, you can have the same. Gets you B blood type. Actually, let's move this a little bit further down. Now you can have AB, right? So if you have both dominant alleles together, right, as a heterozygote, but with 2 dominant alleles. So this is, it's not compound heterozygosity, which is what we talked about with diseases. I think maybe with the other class, but it is a heterozygote, but yet it displays both dominant phenotypes. And then O is easy. O is just homozygous recessive. And that gets you the only recessive phenotype. All good so far? There's the chat gone. There we go. Now, how does that work? And right, if you remember when we started talking about this stuff on Wednesday, one of the things I said is that the scale at which you look at a phenotype kind of determines how you view its inheritance to some extent, right? And especially with very kind of molecular cell level phenotypes, you're much more likely to see kind of odd or intermediate forms of dominance, right? Codominance or incomplete dominance. And so the mechanism behind this is really interesting. I'm going to draw. Oh, you know what? I could use a red pen. Because it's red blood cell, right? That's supposed to be red blood cell. It's not a donut or a large O. And so if you have the A allele, you produce the A antigen. I think it's a glycoprotein. I'm not 100% sure, but it's a protein on the surface of your red blood cells. Right. Now, if you have the A antigen, you have the antibodies to the B antigen. So you have the opposite. You obviously don't have antibodies to your own antigen because then you just kill yourself, right? So you have B antibodies. That's short for antibodies. Right, so if you have whatever antigen you have the opposite antibody. Make sense? I'm going to sneeze. I used to make my daughter cry when I sneezed when she was younger. My eldest, anyway. It's kind of sad. Okay, thank you. Similar kind of deal for B allele, right? You have B antigens and A antibodies. Ah, okay, so no antibody because it's a non-functioning version of this gene. So antigens are antigens, obviously, because they elicit an immune response. And you can change a protein in different ways and it still function, but it'll be recognized as a different antigen, right? So antibodies raised against one will not. Yeah, as far as I'm aware, I don't actually know what genes these are or even what proteins they produced. Yeah, so A and B are both functioning proteins but have a different immunogenic profile, like how their shape essentially. And O is non-functional, it's the recessive non-functioning allele. It's very much like, you know, for example, the spike protein in COVID, right? Essentially, there are multiple alleles or what they call variants of the spike protein. And so the spike protein still functions because any mutations that get rid of its function stop the virus from succeeding, essentially. But they are recognized differently by your immune system to varying degrees. Right, so that's essentially what these are. These are proteins, some sort, on the surface of the cell, which are recognized by another person's immune system if they have antibodies against these antigens. And we'll get to the consequences of that in a little bit. Actually, we kind of skipped ahead a little bit earlier, but you'll see what I mean. So again, the same for AB, but now because you have both alleles, you have both proteins expressed on the surface of that red blood cell. And because you have both antigens present, you can't have any antibodies, because if you have antibodies present to either A or B, your blood will stick together and you'll die, right? So that's why people who are type AB are universal acceptors, right? So they have no antibodies to either of those antigens. So it doesn't matter whose blood you put in them, they'll be totally fine, right? They won't reject it or have issues. Oh, and another point to note as well is when you donate blood, if any of you have done that, your blood is then split after that, separated into red blood cells and plasma. And plasma, depending on, you know, might just get junked, but might get used. But the red blood cells obviously are kept and then put into someone else. And so by doing so, the antibodies stay in the plasma. So that's how you can take B blood type and give it to other individuals, right? Because you don't carry over the antibodies that person had. Now for type O, right, you have two non-functioning alleles and A and B antibodies. So this individual can give blood to anybody because its blood will not, the red blood cells will not be recognized by anyone's immune system, whatever the blood type they have. That's why type O, a universal donor, very handy to have around. But they can only accept blood from other type O individuals because they have antibodies to both A and B. And so any A, B, or AB blood would be recognized by their immune system. So this is how you can express two different phenotypes at the same time. This isn't an intermediate. It's not incomplete dominance. It's not that A is more dominant than B, or so they're equally the same. And so this is how you can have two dominant phenotypes expressed at the same time. It's not super common, particularly with larger kind of type physiology, but it is very interesting. That makes sense, everybody? Oh yeah, agglutination, there you go. Yeah, it's a fun one to talk about. It's and also because you can then, I love trying to figure out, actually I need to ask my dad what my mum's blood type was, because she gave over 100 pints of blood during her life for the British blood bank or whatever that was called. So I'm sure he knows what it is. But anyway, you can work out genotypes. Oh yeah, don't get me started on the rhesus factor, Daniel. I've tried understanding that several times. It's much more complex, and it's actually not completely understood. There are a couple of competing models as to how it works. So I just quietly push that one to the side. So for example, with in terms of lineage, I'm B positive. So I'm either BB or BO in terms of my genotype, right? And so one of my parents had to have a B. So either my mother or my father is either B or AB, right? And as to what else they could be, well, the other one could be, yeah, B, could be all of them actually. Yeah, other than a homozygous dominant. So anyway, it's kind of fun. You can kind of work things out if you know that information. Okay, so a few more things to wrap up from the incomplete, non-Mendelian. Sorry, my brain is fried this week. I don't know what's going on. Go away. There we go. Back. Get out of it. One day I will get a computer monitor. Where are we at? Down there somewhere. Okay, so things get even weirder and wackier, right? In terms of non-Mendelian inheritance. Oh, let me take a picture of that before I forget. When you start thinking about not genes or gene products, right, proteins and stuff, working in isolation, but when they start working in pathways, right, when you have multiple proteins working in sequence or maybe sometimes in parallel to affect an outcome, right? And so here you get a weird sort of effect where instead of having alleles that are dominant over other alleles, now you can actually have genes that are dominant over other genes, right? So essentially, and this doesn't affect how alleles are inherited, right? So the law of independent assortment, the law of segregation still hold true, at least in the sense of what alleles you get in what gametes. But the phenotype from one trait can mask the phenotype for another, right? And so essentially what this means is that you're not going to see some phenotype combinations, right? And also if you do the classic dihybrid cross, so again, this is why it's important to understand the dihybrid cross. If you do that, you will not get the classic 9 to 3, 3 to 1 ratio, right? So essentially this is called epistasis or epistatic interactions, right? So essentially genes interact, which you'd expect them to, right? They don't work in isolation, based on function, right? And so there are many different types of epistasis. This one here is an example of negative epistasis. Right. And typically in terms of negative epistasis, that usually is a result of genes being in the same pathway. Right. So what I mean by this is just an example here. You know, you have multiple enzymatic reactions, but you only have one outcome at the end, right? As a result of all those different reactions, you get pigment, for example, or you get a metabolite that you need or something, right? After that series of events, right? So all of the genes, all of the proteins in that pathway have to function to see the end product, right? And so now, if you have a recessive phenotype for any one of those, or homozygous recessive for any one of those, essentially you have the recessive phenotype. So now there are only two phenotypes, working, not working, right? And when you look at this, let me blow this up a touch, right? You do the standard dihybrid cross. This one's a little bit kind of funkier because it's each parent has kind of the opposite. So you're homozygous dominant for one, homozygous recessive for the other, right? And so both parents display the recessive phenotype, but when you cross them together, you get heterozygotes, sorry, itchy nose, which display the dominant phenotype, purple, essentially. Now, if you remember from dihybrid crosses, there are 4 different categories, right? Essentially, dominant is always 9. Recessive is always 1 out of 16. And then the 2 in the middle is going to be dominant for one and recessive for the other, or vice versa, right? The reciprocal. However, when these two genes interact epistatically, right? So one is epistatic to the other. What you see now is those two new combinations disappear. Right? So you still have the 9 dominant, or essentially in this case, just dominant individuals, but everywhere you have one of the genes homozygous recessive, you'll see the recessive phenotype. So rather than a 9 to 3 to 3 to 1 ratio, now you get a 9 to 7 because the 3 and the 3 get lumped in with the 1 because essentially they each have a homozygous recessive in them somewhere. Now this is interesting kind of in and of itself, right? Because it tells you something about what's going on, but it's also a really neat technique, right? And so if you are studying on, or you're studying a pathway of some sort, and so all of my research, let's move back over here, is on signaling pathways, or one signaling pathway in particular, which in humans is involved in lung cancer. And in worms, it gives them more vulvas than they need, which is kind of weird. But anyway, you can figure out whether or not genes are in the same pathway and whether or not they interact epistatically using this kind of technique. So you can order genes in a pathway, because essentially you can block the phenotype of one with the phenotype of another, right? So this is a very powerful technique to use to kind of build up a picture as to how this pathway or whatever you're interested in actually works, right? What genes are involved? How do they interact? What order do they work in? Things like that. And funnily enough, I actually got some comments on a review of a little paper that I submitted. I think I might have shown some of you in lab, basically suggesting I do that for this particular phenotype I was looking at. I was like, yeah, that'd be a great idea. Not going to do it right now, but certainly would like to in the future. And they were suggesting to do some epistasis experiments. All right. Does that make sense for everybody? You all good on epistasis? There are lots of different types of epistasis. There's also dominant epistasis too, which I always think is cool. Dominant epistasis is why you have white cats. So every time you see a white cat, you go, that's epistasis. Dominant epistasis. Oh, I wish I'd checked this before class. No, it is, I think it's 12 to 3 to 1 from memory. Yeah, I think it's 12 to 3 to 1. I could work it out on the board, but it would take too long. Yeah, basically, if you have the dominant phenotype for the white gene in cats, which is all about melanin pigment, melanocyte cell survival, I think. Yeah. You don't see the phenotype for the fur color, so the cats are just white. Whether or not they have, you know, a dominant like black fur color allele or whatever, it doesn't matter. You'll only ever see white cats. You have to have the recessive phenotype for white to be able to see the phenotype for the fur color. Yeah, so it's 12 to 3 to 1, actually, off the top of my head. So yeah, depending on what ratio that you see, then you can actually figure out what kind of epistasis you're actually dealing with, which is also another kind of neat trick. Good question. It's just fun. I love working through these sort of things. I guess that's why that's the sort of work that I do for my own research. Okay, so another thing that you can also do, oops, that's super blown up. And this is really more of again, more of a technique than anything. So essentially, if you've done a large mutagenesis screen, right? So you screwed up a bunch of animals or plants or whatever, and you've got a whole bunch of mutants that display a phenotype that you're interested in. This is a very classic, what's called forward genetics approach. Then you do a whole bunch of work to make sure that they have just the alleles for that mutant or mutation in them, right? So there's none of the other kind of cruft or junk that you might introduce through such an approach, because you generally screw the animals up pretty solidly. And then the question is, well, they all produce the same phenotype. Is that because all of these alleles are in the same gene? Or do I have alleles in multiple genes which are involved in the same process? And then you go back to the epistasis approach to figure out how those interact. Right? But before you do that, you have to figure out, okay, how many of these are different alleles of the same thing versus different alleles in different genes, right? That's a really important question to ask. And so the way you ask that question is by something called complementation, right? And so the idea here is you're going to cross two different mutants together, and depending on the outcome, you can tell whether or not they're in the same gene or not. So essentially, if they're in the same gene, right, so A and B are different alleles of the same gene, if you cross those two homozygous recessive individuals together, what you'll end up with is a heterozygote, right? Actually, I haven't mentioned this to you, but this is called compound heterozygosity. Funky words. I love funky words. Language is such a fabulous thing. Being able to describe and explain yourself in different ways is just awesome. So, heterozygosity. So that's essentially two different recessive alleles equals recessive phenotype. Right, so whenever I said heterozygotes always display the dominant phenotype, well, I was kind of lying a little bit. Yeah, so the alleles in the top half are A and B, so just different mutations in the same gene, resulting in a non-functioning gene. So if both of them are non-functioning, it doesn't matter that it's a heterozygote, you're still not going to have anything working. And so now that's essentially telling you, okay, these alleles are in the same gene. And this compound heterozygosity is actually a pretty big issue for blue line is the chromosome, Caitlin. And the letter represents an allele that's different between the two. So if the alleles are in the same place, when you put them together, essentially you'll have a double recessive, and then you'll have the recessive phenotype. Even though they're heterozygous, this is the one at the top of the slide. That makes sense? So gene, I don't know what gene would be. Let's say cystic fibrosis, right? Just as an example. So cystic fibrosis receptor is involved in pumping ions, which helps water go out, which increases mucus, watery mucus in your lungs. So let's say upper half A and B are both alleles of, or recessive alleles rather. Which is CTFR stands for cystic fibrosis receptor, I think. Yeah, I think it's receptor. Right. So when you have the AB genotype, you get the recessive phenotype. Now, in the lower half, A and B are recessive alleles in different genes. E.g., A is in CTFR and B is in, I don't know, aquaporin, let's say, water channel. So now if you have both genes of which are involved in the cystic fibrosis disease, right, if they're not working. So now you have a situation where you are homozygous recessive for one, but not the other. And the other individual is the reciprocal, the opposite. So when you cross these two together, now you have a classic heterozygote for both. Right, you have one chromosome from one individual which is recessive dominant, and the other from the other individual which is dominant recessive. All right, and so now you're heterozygous in the conventional sense for both of those. So now you restore function. And so if you see a wild type or dominant phenotype after crossing those two homozygous recessive individuals together, that tells you that those alleles are in different genes. Right? If you get the same mutant phenotype, then they have to be alleles in the same gene. So did that help, Caitlin? Cool. Yeah, it kind of helps to have all of it, you know, in front of you first, because just one bit of it on its own doesn't help. Actually, just another fun fact. I sound like my eldest daughter. She's always telling me fun facts, like funky. Don't know where she gets this stuff from. But anyway, I think, Caitlin, it's not so much the basics. It's I think, well, I mean, maybe, but it's how you think of these things. And so this is one of the shortcomings I have, right, as a teacher in some ways, is that this stuff just, you know, it's like bilingual person speaking a second language kind of thing. So for me, it's just straightforward, but that doesn't necessarily mean it is for everybody else. So that's why asking questions really helps, because that gives me opportunity to go, oh, hang on, maybe this isn't quite as obvious as I thought it was. Right. So I'm not sure it's so much about the basics, but kind of learning to view stuff in a different way, right? Through the lens of genetics, essentially, right? Because once you do that, it's like once you see what you're looking at, you can see it, right? It's the getting to the point of being able to see it. That's the tricky bit. So a good example of this, or fun example actually, is axolotls, because who doesn't love axolotls, right? I mean, they've got cute gills and everything. Actually reading a really weird book at the moment about a sea monster. I don't know. It's one of the ones that was in my Kindle library that has like these huge gills on the outside. It's called Steve. Anyway, axolotls are a type of salamander, and they're super funky because they are what's called pedomorphs, right? Which means that they display the juvenile characteristics as an adult. So it'd be like a frog that looks like a tadpole, essentially, but a big one, which would be a little scary. So there's a very closely related other tiger salamander, right? Which doesn't look quite as pretty. I guess it depends on your whatever floats your boat, really. But anyway, when you cross these two individuals together, right, you obviously get a sterile hybrid, but the heterozygote is normal, right? Does not display the mutant phenotype, the pedomorphic phenotype. And so that tells us that the genes that are responsible for the pedomorph phenotype in tiger salamander, pardon me, or this one, are completely different from the ones that control it in this one. So when you cross those together, essentially you have a heterozygote for all of those genes, and you don't exhibit the juvenile characteristics in the adult anymore. Right, so that's kind of another way of looking at it, essentially. Okay, so we've got a few things to wrap up, and if we got time, maybe we'll get on to at least introducing linkage, right? Because that's something that we really need to get cracking on. It's a pretty chunky topic. Okay, so another kind of extra wrinkle, right? And I told you there's a whole bunch of these different things, essentially, which break Mendel's laws, right? And again, can't help but emphasize that Mendelian genetics does not explain much of real life genetics. It's just a very useful framework to compare all the interesting stuff to and see what changes, right? So most traits that you'll ever encounter as an individual are non-Mendelian. And that's very much the case for human traits, right, which are very complex. So not going to get into too many examples with this, because again, we're kind of getting a little short on time. But something that's also super funky is that you can have what's called variable expressivity or incomplete penetrance. And so these are two kind of different shades of the same color, essentially. So variable expressivity, and these are basically, these are all in individuals, both of these, that have the same genotype, right? So with variable expressivity, the degree to which you express the mutant phenotype varies. So one individual may have a very severe mutant phenotype or disease phenotype. Another individual may not. So this isn't really something that applies much to kind of like infection and disease potentially, or infectious diseases. So COVID is not a good example here. But it is a good kind of example that comes to mind would be like mitochondrial disease, right? Some individuals, they just have like chronic fatigue and, you know, difficulty doing like long bouts of exercise. Other individuals never make it to adulthood, and yet they have the same genotype, right? So this is called variable expressivity. Mitochondrial disease is actually super, super interesting as well. On the other kind of shade, you have incomplete penetrance. And so again, this is where individuals have the same genotype, but some don't express the mutant phenotype at all. So rather than there being kind of like a spectrum where going from mild to severe, you either have got it, don't got it, right? And that's called penetrance, right? So how thoroughly the mutant phenotype penetrates the population, I guess is one way of thinking about it, is the penetrance, right? And so if some individuals do not display that phenotype, then it's said to have incomplete penetrance. One of the classics in the work that I do is a technique called RNA interference, where you knock down gene expression. That suffers severely from incomplete penetrance, right? So you'll get a kind of a wide range of outcomes from the same experiment. Now, one of the easiest things to say is that most traits in populations are complex traits. So complex traits have kind of two elements to them. So they're, how do I put this? That's probably the best way of putting it. So there's kind of two aspects. One is that almost all traits have more than one gene influencing them. So eye color, for example, multiple genes, hair color, multiple genes, whether or not your earlobes are attached to your head, multiple genes, right? Tongue rolling, finger crossing, et cetera, right? All of those are polygenic traits. And also, many of those interact with the environment as well, right? And so a classic example is height, right? So height is clearly height cannot be a single gene or even a two gene trait, right? There's just too much involved in development and body plans and yeah, so on and so on and so forth, right? Limb extension, things like that, right? So there's a clear polygenic genetic component to height. But you can also look at height over time, right? And so, I don't know, old in US is like 200 years old. But I used to work when I was a teenager in a pub, which was quite fun. That was built in, I think, the 1400s. Yeah, give or take. So about 600 years before I worked in it. And I'm quite tall. And every shift, I would hit my head on one doorway or another, sometimes more than one, right? Because when that pub was built, people were short. And they weren't short because genes controlling height have changed that much in the past 600 years. It's because nutrition has, and disease and stuff like that. And so how good your nutrition and your disease burden in terms of parasites and bacterial disease and stuff like that is when you're growing. Hang on a sec. Determines your overall expression of that phenotype. So that's a gene by environment interaction. And so kind of a classic characteristic of a complex trait is a normal distribution of the phenotypes, right? It's not going to be tall or short. It's not even going to be tall, medium, short. It's going to be a continuous distribution with a normal. Most people are going to be kind of average height, essentially, right? So that's a pretty classic example. And most traits are polygenic, and many of them interact with the environment too. Obviously, your eye color does not, right? So it doesn't matter whether you're dropped on your head as a kid or not. You're still going to have the same eye color, right? Either way, or thrown into the ceiling like I was by my dad. That might have caused other issues, but certainly didn't have caused problems with my eye color. And so another fun thing is as well, this plays out in breeding, right? This kind of gene by environment interaction. And so in the US and Northern Europe and the like, crops have been bred for a long time to be much more higher yielding, right? But also the environment they're in is generally fairly stable, right? Predictable rain, plenty of nutrients from fertilizer, things like that. And, in Texas, at least, plenty of sunshine. So you can optimize the population that you're working with, the strain that you're developing, to be very high yielding in ideal environments. However, in poor environments, poor quality environments, so drought or nutrient limitation or stuff like that, those you'll see the trade-off, right? Because there's always trade-offs, right, in breeding. You can actually see that in dog breeds, right? If you breed for like the perfect Labrador, most likely it'll have hip dysplasia by the time it's 8 or 9 years old, right? Where their kind of hips basically fall to pieces. And it's the same in crops. There's always trade-offs. So if you kind of push towards one end of the spectrum, you give up something at the other. Now, if you go and look at kind of crops in Africa or India, even animals like cows, for example, they tend to not be as high yielding, but they're much more robust, right? And so when the environment deteriorates, which it does frequently in Africa and India, depending on time of year, you're still going to get a reasonable yield from that. Actually, fun example, just kind of popped into my head. Parasite resistance in sheep. So I knew a guy who spent years and years and years and years trying to breed sheep that were resistant to parasites, right? Because that parasite burden reduces the yield and health of the sheep, as one would expect, and essentially found that you never could, because you could get parasite resistant sheep, but they would be small and scrawny, right? And they would never grow particularly large. Or you could get nice, plump, large sheep, which make great roast dinners, but they will be susceptible to parasites, right? So there are always trade-offs whenever you kind of mess around with these gene by environment interactions. It's just, fun to talk about. Anyway, that's that for this part of non-Mendelian inheritance. And ever so quickly, almost so I can just say that I have, which is a little silly. Let's get rid of that. We need to make a move on linkage. Right, and so I just want to introduce the concept. Some of you have already had this in lab, which is great. Anyone that hasn't will get it next week anyway. But I want to introduce this topic because it's a complex one and there's a lot of math involved. And it's, again, one of those things that, once you get it, it's real easy. It's getting it, which is hard. That's, I guess, like most studying, really. So the crux of the issue here, right, is really most genes interact genetically if they're on the same chromosome. So we've been dealing with this kind of classic Mendelian viewpoint where the two different genes that we're interested in are on different chromosomes. Those chromosomes are sorted independently. You'll always get an equal quantity of all the possible gametes, right? However many you're looking at, different genes, they all act independently, so you will get an equal number of each of them. Now, if you look at this, right, so this is a human chromosome. You see the P and the Q, the petite and the whatever Q stands for. Some of these genes are very far apart, opposite ends of the chromosome, whatever these are. Over here, we've got one called Wnt, which is a signaling gene. That's the only one I recognize from there, on the salamander one. Some of them are very, very close together, right? And so linkage is all about distance between genes on the same chromosome and the probability of recombination occurring between them. And we'll go back over this again on Monday. But essentially, the deal is if those genes are on the same chromosome, you have two possible outcomes in terms of recombination. One, crossover may occur between them, in which case you'll get 4 different possible combinations of alleles. This is from essentially a heterozygous individual for both genes. If recombination does not occur between them, and that's because those two genes are fairly close together, you will not see the recombinant chromosomes. You'll only see the parental non-recombinant chromosomes, right? And so now you have an unequal number of possible gametes with the different combinations. That's linkage. So we'll go through this in much more detail on Monday. I would suggest, if you have time and inclination over the weekend, have a read ahead through this PowerPoint so that some of these terms and ideas are at least familiar. It'll make life a lot easier for you. Cool. Ta-da! Can't believe I even got to talk about parasites. Parasites are awesome. Most highly evolved creatures in the world, as far as I'm concerned. Anyway, on that note, have a great weekend, everybody. Study hard, have fun, enjoy the cooler weather, which is a blessed release or relief, maybe one of the two. And see you on Monday.